So, does that work? Okay, we're rolling. Well, first, my thanks to Michelle for taking over while I was gone. I had a good time going back to a wedding and visiting family, and I appreciate the opportunity to do that. And the whole crew was pitched in. It's been great, so thank you all. Now you're back to reading my scrawl as opposed to Michelle's great handwriting, but here we are. What we're going to do today is we're going to start a new section. All right, the section we're going to start is the nervous system, and I want to introduce this as the nervous system as a machine and tell you some stories about how we got to this idea of thinking of the nervous system as a machine. And we can think about the nervous system really as a three-box model, and anyone who has done any computer science will recognize this as data in, analysis, and data out. Basically, that's what the nervous system is doing. It's taking information in from the peripheral nervous system from the outside world. It's analyzing that information. It's integrating that information. It's making a decision, and then it does something about it. Now, how that works is largely a mystery to this point. We have some ideas, but how we take information in and how we really analyze it and make a decision and then do something about it, that black box, which we're going to talk about as the central nervous system, is the brain and spinal cord, that black box is still largely a mystery, and we spent the last 170 years trying to sort this out. We'll see, though, that on today's discussion, we can break down the nervous system into three basic parts, and we'll refer back to these ideas over and over again. First is we have a peripheral nervous system. This is out in the periphery. Now, I'm going to oversimplify this. I'm going to talk about sensory input. If you take a neuroanatomy course, you'll see that this isn't quite right, particularly for vision, because the retina is part of the central nervous system. But I just want to think about carrying information from the peripheral environment into the analytical unit. So we're going to talk about sensory afferents. Afferent comes from the Latin. It means to carry toward. And then we're going to think about a little bit about what we have in here in the brain and spinal cord. We're going to spend some time on this, even though there are many other courses that you can take that will go into much, much greater detail and depth about how this analytical unit integrates information, makes a decision, and then that decision has to be carried out to do something. And for the most part, when we think about carrying out to do something, we think about motor activity. We activate muscles. And so there's going to be the peripheral nervous system, which is motor out, motor efferents. This term efferent, again, comes from the Latin. It means to carry away. We're going to tell the story mostly from the point of view of muscle activity. But recognize, too, that we could talk about this in terms of the autonomic nervous system, and it, too, will have a motor efferent out to affect glands and smooth muscle and cardiac muscle. Now, we can't spend a whole lot of time on sensory transduction on here. In this class. We just don't have time. But if you're interested in this topic, you can take other classes, particularly a cellular neurobiology course, BMS 325, and they go into great detail about how this, in fact, is carried out biologically. All I want to convey to you today is to say that in sensory transduction, we're changing one form of energy to another form of energy. And if you sit back and think about it for a moment, you recognize that everything we know about the world around us is coming in through a very small number of forms of energy. It comes in through electromagnetic radiation, which is, for us, light. Other animals can use different forms of this, and we'll talk a little bit about that. It's mechanical. You can think easily about mechanical input because you're touching things. You can, if you put your phone away, I'd appreciate it. Put your phone away, I'd appreciate it. Thank you. I mean, the phone thing, I'll mention this here now. It's not so much that I care if you're on the phone. That's up to you. My problem is I see you on the phone, and then I'm thinking, why are they on the phone? What's so important on the phone? And my mind goes wandering off, and then I lose track of my lecture, and then I get grumpy. And so I'll just ask you to keep your phones away. So we have electromagnetic information, which is light. We have mechanical, which we can think of easily in terms of touch. We reach out and touch something, we change the form of the tip of our finger, and we can sense that as sensory input. And then finally, this chemical, and this is taste and smell. This is probably the oldest of all of the sensory inputs. If you think all the way back to early creatures, even single-celled creatures, they had to find a source of food. And so they would follow that pathway of chemicals to the source of food. So this is probably the oldest. And we'll talk a little bit about that. But let's first bring up some simple ideas. of how we think the nervous system works. And the first one I'm going to talk about is simply a kind of computer model where we say we have data in, then we have another box, which is analysis, and a third box, which is data out. We can take this same three-box model and apply it to the nervous system, and here we can label it as the three-box model. Of the nervous system. And here we change the boxes just a little bit. The first box now is going to be sensory in. This is going to be part of the peripheral nervous system. This is where we're carrying information from the outside world. To the next box, going to be the central nervous. So where here in the computer model we had data in, here we also have sensory information coming in. That next box leads us to the central nervous system, which is the brain and spinal cord. We do this a little differently. Let me label this the integrative unit. This is where we're going to take sensory information in, and we're going to analyze and figure out what's going on in the world around us. And in fact, this is the brain and spinal cord, biologically. The brain and the spinal cord is the central nervous system. So once we've taken sensory information in and we've analyzed it, then we have to do something about it. And that's the third box here. The third box is motor out. This is, in fact, again, the peripheral nervous system. And we're carrying information. This is where we carry information. Away from the central nervous system, CNS. We term these things motor, efferents. The term efferent comes from the Latin. It means to carry away. So there are these two very similar words which mean very different things in neurobiology. An afferent carries things toward. Efferent carries things away. Now, the way I think about this, the way I try to keep this straight, is efferent has the same word root as to effect, the verb effect, to bring something about. And so the motor efferents are going to bring something about, particularly going to bring about movement. You can sort of normalize all this and think about it in everyday terms. And imagine one of our ancestors out on the Serengeti plain, and the ancestor sees a lion. And that's sensory input, that's vision. But our ancestor may also hear the lion. They may hear them growling, or they may hear them coming. If the lion's really close, they may smell it. And if it's really bad, they may taste it. But that information is coming into the integrative or analytic unit. And what's happening is the individual is now looking at the world and saying, is that lion hungry? Is it coming toward me? Is it going away from me? What's happening? And you integrate that information. And if the lion's coming toward you, you integrate and you run away. That's the motor out. And that's what we're doing all the time. Our nervous system, in ways that remain unclear, as we're sitting here, as we do everyday tasks, we're taking in sensory information. We're analyzing that information in our brain and spinal cord, and then we're doing something about it and sending information out to carry something out. This is sometimes known as the final common pathway. Now, this simple three-box model is, of course, a far, far great oversimplification. But in fact, these three ideas are the way the nervous system works. And more importantly, we can now think about the nervous system as a kind of machine. It's the first step in taking the magic out of the nervous system. And this is a concept that's been going on, for probably 170 years or so. And the story starts, the way I want to start this story was 1848. And there was a guy named Phineas Gage, and Phineas Gage was working on a railroad crew. And if you know anything about our American history at that time in the early 1840s, 1850s, there was a movement to lay out railroad beds. We're trying to tie the country together with railroads because they'd invented locomotives, they'd invented steam engines. And they found ways to take steam engines, put them on tracks, and move them along steel rails. It changed the entire world. Because now you went from a 3-mile-an-hour world to a 30-mile-an-hour world, an order of magnitude faster. So there was a great push to lay out road beds. But if you think about it for a little bit, you recognize that if you have steel wheels of a several-ton engine on steel rails, you can't climb a hill that's very steep. So one of the major advances in that part of the century was to develop surveying. So they could survey and they could make road beds and make them level and give them a certain gentle incline so that the locomotives could climb that incline. And one of the jobs in the 1850s, 1840s, particularly in areas that were hilly, was to basically blow up rocks and level the landscape. And Phineas Gage was working in Vermont. If you've ever been to Vermont, you know it's hilly and you know it's rocky, lots of granite. So his job was to go out every day and drill holes in the rock. Then he'd put a fuse in, he'd drill a hole, usually several inches deep. Then he would put in a fuse. Then he would put on top of the fuse black powder. And then what he was supposed to do on top of the black powder, he was supposed to put sand. Because that, by tamping in, and then take a rod and tamp in the sand, that forced the explosion of the black powder down into the rock and broke the rock. The problem was Phineas Gage was having a bad day, apparently. Got distracted somehow. And when he took his steel rod, it was just about like this. It was about 4.5 feet long. It had a point at one end, and it was about 1.25 inches diameter. And he started tamping on the rock, and he struck a spark. When he struck a spark, because he'd forgotten to put in the sand, it blew up and forced the rod up through his left cheek and out the top of his head. Now, this is a pretty serious accident. I mean, he dropped like a stone. The rod landed like 20 yards away. But amazingly, he survived it. And the story is probably exaggerated. But so the story goes that he stood up, he woke up, they put him on a cart, they hauled him about 3/4 of a mile to an inn. He sat on the porch, and the doctor came up. The doctor was called, the doctor came up and looked at him, and Phineas Gage looked at the doctor and said, "I've made a big business for you today, Doc." And sure enough, he had a hole in his head, and the doctor went in and fished out a few pieces of bone, stuck his finger in to see where the damage was. At that point, Phineas got sick, and he vomited, and a teacup full of brain went out of the top of his head. How this guy lived, I don't know. It's astonishing. But he managed to survive, and if you read the original accounts, the doctor later. And so he bandaged him up, lanced the abscess, and Phineas, to the day he died, his face because of the abscess. But Phineas survived. Now, here's where the exaggeration comes in, because at the time, people said Phineas Gage was never right after that. But we know that he survived the accident. We know that he toured around New England for some time. We know he went to South America. We know he had a job in South America. We know that he came back to family in 1860 to San Francisco, where he died. He died of epileptic seizures. And we now know that if you damage the brain, and certainly there's a lot of brain damage, that the chance of having epileptic seizures later on is pretty high. In fact, the story is, if you have a skull fracture with intracranial bleeding, your chances of being epileptic in about 2 years is about 40%. So it's not surprising that Gage had epilepsy, and it in fact killed him. But after he died, and the news came east to his doctor, the doctor said, "I want the head." And so they disinterred Phineas, took his head, and shipped the skull back to this doctor in Vermont. And to this day, the skull is on display in a museum in Boston. And the reason this is such an important watershed in neurobiology is probably because Phineas' behavior was changed as much as we'd like to think. But the belief at that time was that Phineas Gage had damaged his brain. Brain changed his behavior. The story is, before that time, he was a guy who loved his family and got along with everybody. After that, he was a difficult human being. Probably isn't entirely true, but the point of this is that it changed the idea, it changed the philosophy of what the brain was. It was one of the first demonstrations that we are who we are because of our brains. Up to that point, we're largely who we are because of our souls. So it's a philosophical change. And that change is something we've been working at for the last 160 years, trying to figure out how the brain works. And we've made a lot of progress. We've done a lot in terms of understanding how brain functions. And I was trained as a neurobiologist, so I'm most interested, I will admit, in this part of the course. But having been trained as a neurobiologist, I also worry sometimes of the Pandora's box we've opened. Because the more we know about the central nervous system, the more we know about the pharmacology, the more we know about the neurotransmitters, the more we're going to be able to regulate and control the brain. That means we're going to be able to regulate and control behavior. Now, along the way, we're going to understanding how the brain works, we're going to discover lots of things that are going to make life better. I think we'll probably be able to discover problems with Alzheimer's disease. Any number of degenerative diseases, I think we're going to be able to solve because we understand the brain. But there are also societal implications. We're going to, by pharmacology, we can change people's behavior. By understanding the genes that are turned on to make one person smarter or better at interacting with other people, we'll be able to design human beings. That's a scary thought. And what happens if the wealthy are able to design their children? Who doesn't want to have a bright, athletic child? And so we begin to see this evolution of the possibility of, because we understand how the brain works, we can then make changes to the whole society. We see it even now. There are situations with drugs, and you know full well, you have examples of this in your own life. You know you're going to come to the end of the semester. You know you won't have divided your time up particularly well. You're going to get to finals, and you think, I have too much to do. How am I going to get all this done? And you think, somebody out there has some Adderall. I'm sure I can solve this problem. And sure enough, it'll help you focus. It'll keep you awake. But then there's this simple ethical question. That's a performance-enhancing drug. And we've decided that in athletics, we shouldn't have performance-enhancing drugs. What are the rules about performance-enhancing drugs in academics, in the competition in academics? On the other hand, if my surgeon's been awake for 20 hours and I'm on the table, I hope he has some performance-enhancing drugs on board. So where do we put these boundaries? And I think that that's one of the That will be a major question in your lives, because we're going to figure out a lot in your lifetime about how the nervous system functions. And in so doing, they're going to raise a whole manner of issues about how we control people's behavior and how we control the genetic descriptions of our children. And in a democracy, we need to be making these decisions ourselves. And I think it's going to be one of the real challenges. And there's some days I wake up and I wonder, should I have ever even entered this business, because we've opened such a Pandora's box. But in the end, for me, it's the curiosity thing. The idea that we can actually ask the question, as human beings, of how our brain works is a quintessentially human thing to do. No other creature on the planet can step back and say, how does this piece of protoplasm work to make me who I am? And we can do that. And that's a fundamentally human thing to do. It's as human to me as the Hubble telescope looking back into deep time. It's philosophically an interesting thing to do. And in approaching this philosophical question, we're going to solve any number of diseases in our society. And I think it's something we need to do. But watch out, because there are certain implications of what it's going to do to behavior. But to me, this is all falling out of this idea of the nervous system as a machine. If you don't think of the nervous system as a machine, you don't think of it in terms of something you can manipulate and change and understand. So I come down on the side of I want to know how it works, but there are certain risks to it. So let me go on here, though, and talk a little bit more about sensory transduction. Again, we don't have time to get into all of the molecular details of sensory transduction, and these things are well understood. If you take a cellular neurobiology course, you'll get down deep into the molecular mechanisms of sensory transduction. But I just want to introduce you here to a few ideas, a few general ideas that are universally true. And that is, to start with, in this theme of the nervous system as a machine, is to say that sensory transduction uniformly is a change in energy form. To the movement of ions across a membrane. In order to transduce, to change the form of energy and then recognize that change in the form of energy in the world around us, we have to change that energy form to the movement of ions across membranes. It doesn't matter whether we're talking about the three major forms of energy. All of them will boil down to the movement of ions across membranes. Now, it's a bit of a surprise to me to think that everything I know about the world around me comes in from three major sources of energy. One is electromagnetic. The second is mechanical. And the third is chemical. The electromagnetic one for us is primarily light. It's light and vision. Now, our ability to sense the electromagnetic spectrum is pretty limited. There's an enormous spectrum out there. It runs from very, very short wavelength, very short wavelength energy like gamma rays and X-rays, all the way up to very long wavelength things like radio waves. And along that spectrum, all we can see is from about 375 to about 750 nanometers. 375 is blue or violet, and 750 is red. One of the things I find interesting is that not all animals, all creatures, see in the same range. So, for example, if you think about a honeybee or other pollinating insect, they can see down into the ultraviolet. They can see down, 350, 340, 320. And it's an interesting thing to do if you go take a camera which has a CCD chip in it that's sensitive in the ultraviolet, and you begin to take pictures of flowers. You recognize that there are whole new patterns of decoration in those flowers. And the reason for that is that those flowers are attracting pollinating insects, and they can see down into this 325, 350 range. We can't see it. So there's if you take a picture of a flower using an ultraviolet sensitive camera, you'll see whole new patterns. The other side of the spectrum is once we get above about 750, we now get an infrared. And infrared is heat. We're all familiar with that. We can put our hand close to a hot pan and we can feel the heat. But we can't really image with it. But if you look at a pit viper, the pit vipers are the pit vipers are the poisonous snakes of North America. These are things like the water moccasins and the copperheads and the rattlesnakes. And what characterizes them is between their eye and their nose, there's a pit. That's how they got their name. They're pit vipers. But within that pit, there's a localization of the nerve endings in the face of that animal. And those nerve endings are sensitive to heat. And they can, in fact, the rattlesnakes, the pit vipers, can, in fact, image on their sensory cortex. They can image a heat signature from an animal. So if they're out hunting and they find a bird or a mammal late at night, it's dark, they can actually see that creature and have dinner. We can't do that. I mean, it's only been in the last 15 years we've been able to generate machines that allow us to see in the dark. They too are infrared sensitive. But animals figured this out millions of years ago. There are also cases where if we look at animals such as sharks and rays, and you've probably seen these videos on Discovery Planet or Animal Planet, where you see this beautiful animal, a ray, sort of flying above the surface of the sand. And suddenly there's a flurry of activity, and the animal's had dinner. What the animal's done is it's found a creature buried in the sand. And the reason it found the creature buried in the sand is that we all have, because of our nervous systems and our muscles, we all have an electromagnetic field around us. In air, we can't, that electromagnetic field doesn't go very far. But in water, particularly in seawater, it's a conductive medium, and so there's an electromagnetic field around these creatures, and there's some animals that can sense that electromagnetic field and use it to hunt. It's said that a shark could recognize theoretically the voltage in a flashlight battery across something like 10 kilometers. So they're very highly sensitive signatures of these animals, which the shark can pick up, recognize as, and consume that. Now, we're pretty good at this. We can differentiate colors, but we just don't have the breadth of some of these other creatures. Another example is that there's some migrating birds who are able to sense the electromagnetic field of the Earth. We don't know exactly how it's done, but we know that you can take, for example, a homing pigeon, take it out 500 miles from its loft, throw it up in the air, it will fly back in the same day. The reason it can do that is it can navigate, and part of that navigation is to have an inborn compass. The animal has sensed the electromagnetic field of the Earth. And by sensing the electromagnetic field of the Earth, it can navigate its way home. We can't hope to do that. I mean, I can't find the garage without a GPS. And so there are these creatures who have different sensory inputs, who have a different view of the world than we do. And they come from different selection pressures, which have allowed them to develop different tricks of seeing the world and recognizing different aspects of the electromagnetic range. We're limited to about 375 to 750. Mechanical is another form of energy which we can sort out. And I think for us, the easiest thing to think about is touch. You can imagine reaching out and touching something or picking something up. You can feel it. And one of the ways you can feel is you're deforming the skin on your finger. And by deforming the skin on your finger, you're setting up electrical signals. They're going to propagate from the tip of your finger into your analytic unit, into the brain and spinal cord. Make a decision about what to do with that, what it is, and send the information back out again to do something about it. So again, it's the same issue with touch and in light. This is carrying information into the central nervous system where we're going to analyze that information and do something about it. The other one that's mechanical, that's less obvious, is audition. This is hearing. It's a remarkable system. The way there are cells called hair cells in your inner ear, and they have these cilia on them. And each of the cilia are connected by a connection, a very thin biological wire. And then when the membrane that these cells are sitting on, when that begins to move in the response to sound, and this top begins to move, these wires will actually open channels and allow ions to flow through. Again, another example of how we change mechanical movement to electrical movement. Where that mechanical movement is coming from is that as I'm standing here, for example, I'm making sound. As I make sound, all I'm doing is I'm setting up a wave in the air. There's a compression and a rarefied region behind it. That vibration now is sensed by your tympanic membrane. The vibration on the tympanic membrane moves a small set of ossicles. That moves the oval window, which then moves the fluid, which allows ions to come in through into these cells and begin the process of sensory transduction. Then, of course, that information is carried to the brain. It's being carried by the sensory afferents. It's being carried to the brain, and then it's being analyzed. And you're able, in ways that are completely mysterious, to take those sounds and convert them into language. And then from converting it into language, you then take that language and convert them into thought, and then into ideas, into creativity. How that gets done, we don't know. We have the barest of ideas of how it begins here, but how the analysis is done once we get the brain spinal cord is completely mysterious. We'll work it out, but it's completely mysterious at this point. We're pretty good at this because we can distinguish very fine difference in sound. I can't do it, but some people, you can say, make a 440A, and they can sing the note. And that's an ability to take in the information about what it sounds like, create that in the central nervous system into motor activity to activate the vocal cords to make a particular sound. We're really good at that. I'm not, but many people are. And this is because we are so sensitive in our hearing. That's largely because we've needed language. If you take other animals, though, they have a different form of sensitivity in hearing. They're owls. Some owls hunt by vision, but many owls hunt at night. And the way they hunt at night is they hunt by hearing. So that what they're doing is they have a system that they can listen for an animal crawling along the forest floor, a mouse running along the forest floor. And they can hear the difference in timing of the sounds coming to one ear at the other. And then from that, they can image where that mouse is in space, fly down, and grab the mouse. Years ago, they did these experiments where they would have speakers laid out on a floor, an absolutely black, pitch black room. And every time, the owl would come down and grasp the speakers in one direction, exactly where that mouse would have been. There was no mouse. It was just the sound of the mouse running from speaker to speaker. And the animal could hear it and assume that was a mouse and attack it, just by sound. I can't do that. I'm lucky to know where the train's coming from. But these other creatures are able to have these different kinds of use of sensory information, all in terms of how that information is analyzed in the central nervous system. And finally, there's chemical. And chemical really boils down to two. One is a sense of taste and a sense of smell. we're doing here is we're using the shape of molecules to initiate ion movement across membranes. We think that this is probably the oldest of all the senses because you have to eat. And so the earliest of animals could sense molecules in the water column. And then they could sense by swimming up or down the water column, they could get to the source of those chemicals and have dinner. We can still do some of that. We're really much better at combining smell and taste, these two different chemical interactions. Taste is something, of course, we all know happens on the tongue, but there are only a handful of inputs for this. Taste comes as sweet, salty, bitter, and umami. Or maybe one more sweet salt. And you can taste this as those four basic tastes. But to get flavor, you have to mix in smell. And there's an interesting experiment that's worth doing. Go get yourself a bag of jelly beans. And close your eyes, reach into the bag of jelly beans, grab a jelly bean, hold your nose, put the jelly bean in your mouth and begin to chew. You'll recognize that jelly bean is sweet because that's taste. But until you let go of your nose and allow those aromatics to get into your nasal cavity, you won't know what the flavor is. But immediately when that smell comes up into the nasal cavity, then you'll know whether or not that jelly bean was licorice or cinnamon or lemon or chemicals they use to make those flavors. You'll immediately. But you can't do it just from taste. It's a remarkable sort of system. I mean, there's some sommeliers who can taste a wine and tell you where that wine was produced. They can tell you within a few hundred yards of where the wine was produced. I have no idea how that's done. But it's a highly trained and highly sensitive system which comes out of the sense of taste and the sense of smell. So are there any questions up to there? Okay. If not, let's move on. I didn't think I'd get this far, but let's move on and we'll start talking about something known as the neuron doctrine. The neuron doctrine holds that the neuron is the fundamental structural and functional unit of the nervous system. We take this for granted now. This is a basic part of neurobiology. It's built on this idea that the neuron is an individual unit, and it's the functional and structural unit of all neurobiology. But this wasn't always the case. We didn't always believe this. This is an idea that came up in the late 19th and early 20th centuries, and there were two main arguers about this. The two arguments that went on were, first, the cellularists, and the other group were the reticularists. The problem arose here because they simply didn't have the ability in the microscopes of that time to see that they were individual neurons. All they could see were the neurons in the central nervous system. And what they recognized was that in the early days of the histology, the kinds of dyes they had were dyes that had been developed by the coal industry. They were aniline dyes. And they began to use these aniline dyes to see cells in histology. That's why much of the early histology was done in the late 19th century, mid-19th century, because they had these new dyes. The problem was when they applied those new dyes to the central nervous system, they would see cells everywhere because the central nervous system is highly packed cells, very densely packed cells. And so when they tried to sort out what was going on in the central nervous system by cutting sections and looking at those and staining them with the typical aniline dyes, there were just cells everywhere, and they couldn't make out the detail. And so this then led to two opinions. One opinion, the reticularists said that every cell is connected to every other cell by cytoplasmic continuity. We'll call them cytoplasmic bridges. The cellularists said, no, no, no. The nervous system is made up of individual cells called neurons. And each neuron is separate from its neighbor. This argument went on for several years. And it turned out, I'm going to have to go to the upper panel. It turned out that largely it was solved by a change in the staining techniques, where the aniline dyes that we talked about, such as the Nissl dyes, could state cells by their nuclei. They couldn't see the extensions of the cell. They couldn't see the processes of the cell. And so there was a fellow working in Italy, and his name was Camillo Golgi. And Camillo Golgi was working at one of the major universities in Italy, at Pavia. He was the center of the biological universe. And he invented a staining technique. And the staining technique used chromium and silver. And it was a revolution, because what it did is it labeled neurons in their entirety. But it only labeled about 1 to 2% of the neurons. Now, at first blush, you would think a stain that only labeled 1 to 2% of the things you wanted to look at wouldn't be very useful. But in this case, it turned out to be really useful, because you weren't troubled by the other 98% of the cells. You could see this 1 or 2% of the cells, and what's more, you could see them as a full structure. Now, interestingly enough, Golgi took that data, took that information, and Golgi, in some ways that are obscure to me, from that said, every cell is connected to every other cell. There's cytoplasmic continuity among all of them. And so he was one of the major proponents of being a reticularist. Meanwhile, there's another guy named Santiago Ramon y Cajal. And Cajal was working in Spain. He started his career as an anatomist at Valencia. He then moved on and worked at Madrid at the major medical schools there. He has an interesting history. He was always interested in art. He loved art. And he wanted to become an artist, but his father said, become a doctor, do something useful. Some things never change. And so Santiago Ramon de Cajal took his father's advice, became an anatomist. But in addition to being an anatomist, he was a microscopist. And what he did is he used the Golgi method. He used the Golgi stain. He modified a little bit, but not a whole lot. And he came to a completely different series of conclusions. He said that the nervous system is composed of individual units. Called neurons. So Santiago Ramon de Cajal is the major proponent of the cellular. Cajal was right. This is the foundation of all modern neurobiology. This is the foundation upon which all of our thinking in neurobiology rests. It's the idea that individual neurons are communicating with one another through chemical synapses. He founded that idea. The other idea that he founded was something called the law of dynamic polarization. That's a lot of fancy verbiage, but what it means is that information in neurons moves in one direction. One of the weaknesses of the reticularist thinking was that there was no way to see how information would move from one place to another, how it would move in one direction. So this was just a gamish of all these cells tied together. So that was one of its weaknesses. And Cajal was the one who sorted it out. And he sorted it out simply by being a great anatomist. What he did is he would go into the lab in the morning and he would sit down and go through slide after slide after slide of portions of the brain that had been stained with the Golgi stain. Then he'd go out in the afternoon. He'd just hang out in the park in the afternoon, and he would feed the pigeons. And in the evening, he'd come back to the lab. And in the evening, he would then draw without tracing it. He just drew it as an artist. He drew what he'd seen in the morning. He was sort of averaging what he had seen from looking at those slides in the morning, and in the evening, he would make these drawings. And until the late 60s, these were the best images we had of neurons. Till we could begin filling cells with dye. These were the best images we had, were Cajal's drawings. And he made this major contribution to neurobiology. And in 1906, there was a sharing of the Nobel Prize. It was shared by Santiago Ramon de Cajal, and it was shared by Golgi. And if you go back and you read the seminars that these two guys gave, Santiago Ramon de Cajal gave this lovely talk, being very gracious about how the technique that he used was developed by Golgi, and that all the thinking, which by 1906, which had culminated in mostly the acceptance of the cellular theory of neurobiology, that he was very gracious in terms of thanking Golgi for his contributions. Golgi, who was being given the prize because of the development of the stain, gets up and gives another seminar and basically says, Cajal has it all wrong. He's an idiot. He completely screwed it up. Now, of course, Golgi was wrong. And to the end of their days, they hated each other. It was this sort of personality clash to the time they died. And Cajal went on, of course, and did really great work in cellular biology, whereas Golgi ended up being influential in the medical schools in Italy. Until the mid-1930s, they were teaching the reticularist theory simply because of his political powers. With that, I'll let you go.